Hello everyone, I am Brigrish Mishra, a PhD student at IIT Bombay. So this is week 9 of the Biophotonics course. This is I couldn't do the lecture on last Friday, so I shifted it to this Tuesday. Okay, so next lectures will be usual basis, that is each Friday 6 to 8 p.m. Okay. So let's start this week 9, which is biophotonics. So mainly theoretical portions has been covered. Some theoretical techniques regarding this laser tissue engineering has been covered. So there is, I mean, again, these are all theory things, so there is no numerical problem. So I'll just discuss the questions given in the assignment. I mean, although since you probably know all the answers, because it was also uploaded in the portal, but I'll just discuss all these questions given in the assignment, and yeah, we will end the lecture after discussing all the questions. So the first one is laser induced pairing of two or more tissues is known as it's tissue welding. So basically binding of two or more tissues, is tissue welding. Okay. So let's discuss more about the tissue welding. uh, so I'll just try to write couple of things here. So in this process, laser is used to join or bond tissues. Okay. Generally, so there are different type of lasers which can be used in this tissue welding, but currently uh, there are only few of them which is used. Only yttria based laser has been used. Anyway, we'll discuss that later. But this technique, this tissue welding technique, is currently only applied to the soft tissues. Okay. So currently this technique is applied only on soft tissues. Okay. uh, Generally either argon ion or double frequency based yttria gallium laser are used for tissue welding. Okay, why is it so? Because they produce deeper and more uniform tissue welding compared to CO2 based laser or any other laser. Okay. Now the important thing is choice of laser. Okay. So choice of laser wavelength and exposure parameter, that means time relaxation, tissue relaxation no, sorry, time.
I didn't get that. Could you try again?
Sorry. And exposure time, thermal relaxation time, yeah. And exposure parameter, which is thermal relaxation time, parameters like energy, pulse, duration, etc. are clearly dependent on the tissue application. That means which tissue you want to weld. Hello. Yeah, sorry, some connection issue was there. So the on the tissue application, optical optical penetration depth, optical penetration depth, and the thermal relaxation time in the tissues to be welded. Okay, so there are basically three approaches. uh, Three approaches. Number one, direct welding of tissues. Number two, laser soldering. And number three, dye enhanced laser soldering. Dye enhanced laser soldering. Okay, so these are some general uh, information and characteristic of this tissue welding. Okay, so next move on to the second question. Most used laser to treat ophthalmic disorder like retinal tears or retinal blood occlusion is this is a C, uh, near visible laser. Okay, so these are also known as thermal lasers, also known as thermal lasers. So these are again most commonly used in ophthalmology. So the why these users are used, that is an important question. So the lasers wavelength is important as absorption is heavily dependent on it, because in order to treat you need absorption. Okay, absorption, because like basically lasers work is heat treatment using heat generation. Okay, so for that you need absorption. Yeah, so absorption on it. So common targets in the retina. Common targets in the retina are such as hemoglobin and melanin. Okay, so these generally absorb wavelength of light between 400 to 700 nanometer. Okay, so this is the important region, that is why it is near visible laser, mostly in the red region, like 5 green or red, but this is the general range. Okay, why it is called near? Because like mostly green or red is used. 400 to 700 nanometer. Additionally, lasers above 500 excuse me nanometer are preferred in the macula as yellow xanthophyll pigment pigment in the additional lasers above 400 are preferred in the macula as yellow pigment uh, in the as yellow pigment in the macula preferred in the macula as yellow pigment because absorption of light happens at wavelengths between 450 to 500 nanometer, and the macular damage and the macular damage can be avoided. Okay, so that is why near U visible laser is used. So the third question is excimer laser, okay, which is predominantly used to treat LASIK, is what laser? It is basically argon fluoride laser. Okay, so these lasers are mostly a mixture of rare gas as well as halogen. Okay, because you need very high energy in UV region for the treatment of LASIK. Okay, So that is why argon fluoride laser need to be used. So the property of the excimer laser that makes its interaction uh, with the stromal surface of the cornea cornea its interaction with the thermal cornea is unique. Cornea is unique compared to other lasers. Okay, cornea the stromal surface of the cornea unique compared to the other lasers is the ability to impart very high energy to such tissue without correspondingly high thermal component. So the important thing is send the very high energy, okay, to these tissues without a corresponding high thermal component. Okay, so that is the main reason behind using this excimer lasers. Okay, now let's talk about something, why this ability, okay, of these lasers. So this is due to the ability of the mixture of rare gas, in this case it is argon, and halogen, in this case it is fluoride, to produce a wavelength of 139 nanometer. This is in the UV region, so that means it contains very high energy. Okay, in UV region, very high energy, which is extremely well absorbed by the covalent bonds such as carbon, nitrogen, and carbon carbon bond found supporting the corneal epithelium and stroma. Okay, so the next question is the amount of time that it takes an object to return to the ambient temperature following heating is known as thermal relaxation time. Okay, so basically thermal relaxation time tells how quickly or how slowly an object loses its heat energy to the surrounding. Okay, so it can be written as TRT in short form. Okay, thermal relaxation time. So TRT tells how quickly or slowly an object loses its heat energy. Okay, it was introduced into laser treatments to put a restriction to put a restriction on the laser pulse duration to minimize thermal damage to adjacent tissues. So thermal relaxation time is very important because when we do the laser treatment we don't know how long does it take to absorb the energy or and convert it into heat. Okay, then again if the exposure time is very high it can damage the tissue. So then scientists uh, fix this uh, thermal relaxation time as a time in which they will expose the tissue with the laser beam. Okay, which avoids the high thermal damage, okay, of the tissue. Thus most of the thermal damage would be confined to the absorbing target and not the other healthy tissues. Okay, that is why TRT is very important. Okay, so let's go to question number 5. Ideal characteristic of laser use in tissue welding should be: uses short pulse laser absorption, selection of laser wavelength to limit absorption, to limit absorption. Sorry, use short pulse laser and then selection of laser wavelength to limit absorption. Use chromophore to increase bonding strength. So all of them are correct. Like these should be the characteristic of the laser used in tissue welding, which we had already discussed in the question first. So just this is the right option. Okay, let's go to the question number 6. Chromophores, which are mainly targeted by lasers for tattoo removal, is melanin. Okay, so for tattoo removal, melanin or the tattoo pigment is targeted. Okay, so in tattoo removal, the target chromophore is melanin or tattoo pigment. Laser light causes extremely laser light causes extremely rapid heating of melanin or sorry tattoo pigment granules. This uh, this fractures these submicron particles and kills the cells that contain them, and in this way the tattoo is removed. Okay, so this is a simple process by which tattoo removal is occurs, but this has to be very uh, precise, so that it should be melanin to target. Okay, not the any other layer. Okay, now the next question is photorefractive keratectomy is advantageous for people who have a thin cornea because it doesn't require cutting the cornea. Okay, so if you have very thin cornea, you should not go for the LASIK. You should go for the this PRK. The short form is PRK. Okay, so so it is a type of refractive surgery which uses a laser to treat vision problems caused by refractive errors. PRK is used to treat myopia, hyperopia, and astigmatism. Okay, now the important part is for patients with dry eyes or thin corneas, this PRK is a good choice over LASIK because it doesn't require to cut the cornea. Also, if you are very active, if patient has a very active lifestyle or has a job which he cannot get leave or it's urgent, then you should also go for the PRK, not for the LASIK, because it doesn't require to cut the cornea. Okay, so that is also a factor, an important thing for the PRK to choose over LASIK. Now question number 8 is, small oil producing gland present in the skin of mammals is known as, so this is the right answer, is sebaceous gland. 8. So what is sebaceous gland is? So sebaceous gland is small oil producing gland present in the skin of mammals. These are usually attached to hair follicles and release a fatty substance which is known as sebum into the follicular duct and hence to the surface of the skin. The glands are distributed over the entire body except palms of the hand and the soles of the feet, they are most abundant on the scalp and face. Okay, so this is the general information about sebaceous gland. So the next question is major advantages of using laser in dentistry. So I think this sir has discussed in detail in the lecture. So all of these are the advantageous property: fast healing time, low chance of infection, less discomfort. So D, all of them. Now the last question is, it has been claimed that low level laser therapy can help in reduce pain, reduce inflammation, both A and B, none of them. So it is both A and B. However, this reduced pain, sometimes it is mentioned that no pain, and sir has also said that these claims are not the general claims. Okay, some have claimed that it doesn't have any pain in the process, but some researchers, some group, some universities deny those things. Okay, so it's like first do the proper inquiry and then use it. So basically use it on your own risk. Okay, so yeah, that's it for today's lecture. We will meet on the coming Friday. Thank you.